Hey, hello everybody. Good afternoon. It's already so dark. Okay, questions here. Professor, when is the final day and time? Can you please confirm? And it is not cumulative, right? Just the nervous system and multiple choice. Yes, so, all this information is posted on our website in the syllabus. So, day of the exam is December 16, which is a Tuesday. And the time is different, 1:45 to 2:45. Okay, so I will post an announcement, but so that you know ahead of time. And I announce it in the first day of class, and it's also in the syllabus posted on our website. So December 16, 1:45, in person in the assembly hall. Okay, so today, we're finally going to discuss the central nervous system and the brain today. But before we start, I want to make this announcement or remind you, any student that missed one of the lecture exams, please send me an email to confirm that you want to take a makeup on the day of the final, right after the final. Okay, so please send me an email. Without that email, I don't know if the student wants to take or not the exam. Okay, so let's move on. The brain is a very, very complex organ, the most complex structure in the whole universe that we know so far. It has 86 billion neurons, 86 billion neurons. Each neuron can have more than 200,000 synapses. So you can imagine the complexity of this, and to be able to study it and to make some sense of all these networks in the brain and what controls what. Okay, so the brain, some of its functions are to register sensations. What does that mean? That means that what happens at the periphery in the external environment or internal environment is going to be perceived by receptors. Those receptors are able to specifically, depending on the modality of the sensation, they can convert it into an electrical current that then is going to be conducted by the sensory neuron and reaches the central nervous system. Can be the brain or the spinal cord, but now we're talking about the brain. So those sensations are registered in the brain, in specific areas of the brain. Then it makes a correlation between, if there's electrical currents that can reach the visual center and the auditory center, and it makes correlation with all this electrical stimulation. What is necessary to generate a response? So it makes the decisions wherever the electrical currents are going to end up in a specific area in the brain. Then a response, another electrical current, is going to be generated and conducted to the periphery to initiate an action. You can't hear me? Am I muted?
I can hear you.
Okay, thank you. Someone can not. So maybe you have your microphone off. Amy. Okay, so all this is to maintain homeostasis and so on and so forth. But then we cannot forget that the brain also has a very important abstract function. So it's the center of intellect. So we can generate new thoughts, new information through the brain. AI, artificial intelligence, is an amazing tool. I use it all the time. And but it cannot generate new information. Its intelligence is based on information that already exists. But it has the ability, it means AI has the ability to scan, to look for information in thousands and thousands and thousands of sources. But our brain is creative, can generate new songs, new poems, and so on and so forth. So the intellect is one of the important functions of the brain. Sophia, can you hear me now? Some people don't hear, some
Yes, I
people.
hear you. Thank you.
Okay, good. Okay, so it's also the center for emotions, right? Be happy, to be sad, to be upset, and so on. And to generate behavior and memory. We talked a lot about memory. So these are the abstract functions of the brain that are very, very important and are also dependent on electrical currents that travel through the nervous system. Okay, so the brain, we can sort of divide it into three levels. So the most inner level that's here represented in this dark red is involved in maintaining homeostasis. And it is shared with other species. So it's dedicated to self-preservation and aggression. So here you have the centers to control heartbeat, the vasodilation, respiration, all these functions that are necessary to maintain homeostasis. They have centers that are represented in this, what we call the primitive brain. We call it the primitive brain because it's shared with other species. It's to maintain homeostasis. Then we have another level of brain that we call the intermediate brain. And this is the center for emotions. Feel happy, feel sad, feel interested, feel bored, and so on. So this is the limbic system. You have to get used to this term. The limbic system is the center for emotion. And you see that the area of the brain that surrounds the primitive brain is dedicated to emotion. And then you see this other brain area that's the neocortex. And that's where the intellectual functions are located. So you can see that it's much, much larger in proportion to the other areas of the brain. So we have the primitive, intermediate, and rational brain or intellectual brain.
Do we have to memorize those names, those long names?
Which long names?
Oh, the pallium, and all that.
No, you don't. An archaeoallium, no. A neopallium, no. Neopallium means brain, neo means new, arche means old, and pallium means intermediate. But no, you don't need to know these Latin names. I can see that you're just focused on what you have to memorize instead of enjoying the knowledge that you're going to learn today. But I understand. How is this such a complex organ? How does it develop? It develops from a very simple structure that is shown here. It's called the neural plate. So it's just a plate, an area, surface that as the fetus is developing, it invaginates and forms a neural tube that has a hollow center. And you see the neural crest also. So the plate, the neural plate, differentiates into a neural tube and to a neural crest. This is the initial steps. Then by the 4th week of embryonic life, this area, the neural tube, now develops into 3 vesicles. The prosencephalon, yes, you need to know this, the prosencephalon, mesencephalon, and rhombencephalon. And these 3 areas are in continuation with the spinal cord that's represented here in purple. So we're going from a flat structure, flat plate, an invagination, a neural tube, and then the neural tube is going to be differentiated into these 3 different vesicles or sections. So all this occurs automatically without us having any control over it. So when we go from 4th week to 5th week, there's more differentiation of the brain vesicles. Now, the prosencephalon differentiates into these 3 areas. The telencephalon, here you see the optical lobes, the hemispheres. So the telencephalon, then that's where the intellect is located. It's going to give rise to the cerebrum. Then the optical vesicles here. The diencephalon, that's part of the midbrain. So that's the prosencephalon. This vesicle differentiates into telencephalon, optical vesicles, and diencephalon. The mesencephalon stays the same, and the rhombencephalon, which is the hind part of the brain, is going to differentiate into metencephalon and myelencephalon. So these here in green are part of what we call the hindbrain, and they are going to be dedicated to maintaining homeostasis. This is where the cardiovascular center, the respiratory center, and so on, where they are located. So you cannot survive without the hindbrain. You can't survive without intellect. Intellect is not required for life. But the hindbrain, the cardiovascular respiratory center, those are essential. So now we go from 5th week of embryonic life to a fully developed brain. And now you see here the myencephalon, and you see the metencephalon differentiates into pons and cerebellum. Pons and cerebellum. This area of the brain is dedicated to fine-tuning movement. It's a little brain, sort of, that is tucked below the frontal cortex. Then we have the midbrain with the mesencephalon, and the forebrain with, so the telencephalon. You can see here the 2 hemispheres, how they grow so much that they go over the other areas of the brain. So here in this image, you see a more detailed differentiation of the brain, going from the vesicles, and then you see the forebrain that is enlarging and growing and growing. 100 days, it's going to surround, involve, hide all the other areas of the brain. So it's going to be major. And this is why we can study the other species and the other species cannot study us, because of the huge size in proportion of the neocortex, the frontal lobe. Here, this area, the neocortex, it grows so much in relation to the other areas of the brain, and it grows so much that it can no longer fit properly in the cranium. So it has to fold many times to be able to fit in the cranium. I find this image very interesting, how all this happens without us doing anything, and reach the 9 months with hopefully a perfect brain with all the different functions. How long is the brain if it's stretched out completely? Oh, that's a very good question. I have no idea. I never thought about this. If you could stretch it all out, it would be very hard to stretch because it's so soft. But how would we can know the number of cells in it, but the size of the whole stretched brain, I cannot answer that question. I'm sorry. I will try to. Maybe someone can look it up quickly. If it was uncoiled, it's a good question. Kevin, try to see if you can find the answer. Sorry, I don't know. Okay, now the complexity of the brain. So it's not only the size, right? So that means now we have 86 billion neurons, right? So, but now the complexity of the synapses, how all these neurons crosstalk, how they form synapses, connections among themselves. So you see that a baby of 5 days of age, so this is a PET scan. Remember what PET scans are? They tell us the activity. So you can inject radioactive glucose that is taken up by the neurons that are highly active. So you see that at 5 days, there is very little brain activity. Everything is blue. So the higher the activity, the dark red represents high activity. So you see neurons, but there are not that many synapses. By 6 years old, the brain is highly, highly active. So you see that 86 billion neurons and many cells, many neurons can receive as many as 200,000 synapses. So one Purkinje cell, Purkinje cells are located in the cerebellum, and they have a very, very extensive dendritic tree or branches. They can receive 200,000 synapses. Will we get any figures, diagrams on the final? No. Maybe some of reflexes or something like that. Okay, so at 6 years of age, our brain is what we call the most plastic. That means it has the ability of forming new synapses. Like it is said that at this age we can learn many languages because the brain is very plastic. The neurons have the ability to form many, many different synapses. Then when we grow to the adult stage, many of the synapses that are formed earlier will disappear, and you have fewer but more pronounced, highly defined synapses. So let's see what it says, that the brain cannot be uncoiled. However, the cerebellum tissue can, which is stated as 3 feet long. Okay, so there you go. Thank you. Okay, so what are the major functions of the brain and their specific areas? So we already said the hindbrain, which is homeostasis, where you can control heartbeat, respiration, hunger, satiety, and so on. So this includes the cerebellum. The cerebellum is where coordination, movement coordination is established. Then we have the diencephalon, which is the midbrain for emotion and sensory. The diencephalon is made up of the hypothalamus and thalamus. So where emotion, the limbic system. And then we have the cerebrum, the largest part area of the brain. And that's where the intellect is located. Okay, brain protection. It's the same layers found at the spinal cord. So instead of the vertebra, we have cranial bones that form sort of a case for the brain. We have the meninges. Remember the dura mater, arachnoid, and pia mater. So the pia mater they're all connective tissues, different types of connective tissues. And the pia mater is the one that's closest to the brain itself. There's a lot of blood vessels. Then the arachnoid is type of a connective tissue that has these projections that make it look like a spider. And remember, the subarachnoid space between the arachnoid and the pia mater, that's where the cerebral spinal fluid flows. And then we have the dura mater above the arachnoid. Then you have bone, connective tissue, and so on, and skin. So for protection, we have bones, meninges, and the cerebrospinal fluid. So the cerebrospinal fluid is produced by the ependymal cells. These ependymal cells are located in the ventricles. You will see that the brain is not all solid. Inside, there are spaces that are in continuation with the central canal in the spinal cord. So the spinal cord the central canal is filled with cerebrospinal fluid that then flows into the ventricles located in the brain. And the cerebrospinal fluid also flows around the brain so that the brain is swimming in this fluid that is located in the subarachnoid space. And the ependymal cells, where are they located? They're located in these organs, sort of areas that we call choroid plexus. And you will see, I will go into greater detail.
Professor, sorry, what was the meninges again?
Sorry?
The meninges?
The meninges. These are the connective tissue. They are in continuation with the spinal cord. It's the dura mater, the arachnoid, and the pia mater. These are the layers of connective tissue shown here also. You see the dura mater. This is the skull. Then you have the dura mater, the arachnoid, and then the pia mater that is in very close proximity to the brain itself. So the brain is opposite of the spinal cord. In the brain, the gray matter is on the outside, the white matter is in the inside. But you see here the meninges. So these are layers or sheaths of connective tissue that protect the brain. And meningitis, what it means is that there's infection of some of these meninges. Okay. So how durable is the dura mater in surgical procedures? So it's tough, but if you have to go through the brain, I mean, the bone is much stronger. You can cut through the dura mater. So what is the gray and white matter? So the gray matter, remember, this is where the cell bodies of the neurons are located. The white matter contains the fibers, the axons of the neurons that are myelinated. And that's why it has a whitish appearance, because myelin is whitish. So the gray matter contains the cell bodies of the neurons and myelinated fibers as well. Spinal cord, white matter on the outside, opposite of the brain, right? So the spinal cord has the white matter on the outside and the gray matter in the inside. Okay, now here, this image shows you the ventricles. So the spaces that are located inside the brain. So you see here the forebrain, and it has two cavities, let's say. We call them ventricles. You see here two ventricles, one on each hemisphere. You see that the brain is separated by a fissure into two hemispheres, the right and left hemisphere. They're not exactly the same. They're not symmetrical, as you will learn. For example, the language centers are, for most people, are located in the left hemisphere. So different functions, the right and left hemisphere have different functions, have many in common, but some that are different. Okay, so let's look now at the different ventricles, these cavities from which the cerebral spinal fluid flows. Here you see this is the spinal cord, and you see the central canal. So blue shows where the cerebral spinal fluid is located. So on each hemisphere, you have the lateral ventricles. Then you see here an intraventricular foramen that allows the cerebral spinal fluid to flow from the lateral ventricle into the third ventricle. And the third ventricle is located at the area, the diencephalon. And then we have the cerebral aqueduct. So this bridge, so these are all hollow areas of the brain, right? So the cerebral aqueduct that conducts the CSF, the cerebral spinal fluid, to the fourth ventricle. And the fourth ventricle is located at the brain stem. So all these ventricles are connected so that the cerebral spinal fluid flows from the spinal cord to the fourth ventricle, the cerebral aqueduct, third ventricle, and then lateral ventricles. But then it also goes through around the brain through the subarachnoid space. So this does not show the whole circuit of the cerebral spinal fluid, only in the ventricles in the brain. So the cerebral spinal fluid, you have around a liter is produced per day, but circulating in the brain, there's around 100 to 160 milliliter. What is the purpose of the spinal fluid? Here it is. Mechanical protection. Why? Because the brain, so between the brain and the cranium, you have the different meninges, but you also have this fluid, and the brain is sort of swimming, floating within the cerebral spinal fluid. So it offers chemical protection. Why? You have a lot of metabolites that can be removed from, and toxins that can be removed from the brain. The cells are producing all these chemicals that can go then into the cerebral spinal fluid and removed back into the venous blood, and also circulation. So it's hard for the cerebral spinal fluid to circulate. If you think about the blood flows throughout the body because it's pumped by a specific organ that is dedicated to the circulation of the blood. That's the heart. The cerebral spinal fluid has no organ dedicated to promoting its circulation. So as we move and so on, that's how it circulates. And actually, I talked about this. There's a kind of massage that's called cranial sacral massage therapy, where you get massage so to promote the optimal circulation of the cerebral spinal fluid. Good question, Kevin. How does the brain gets rid of cerebrospinal fluid. You will see. So it goes through the arachnoid villi and it goes into the venous circulation. You're going to see soon. Here we are. So here we have the spinal cord and the central canal, right? So this is filled with cerebrospinal fluid. Now you have here in blue, so whatever is blue is where the cerebrospinal fluid is. Here you see the subarachnoid space, right, between the arachnoid and pia mater. So you see that the fluid goes through here, and then it comes out to the arachnoid villi. You see here in this area, this is where the cerebrospinal fluid is going to exit the subarachnoid space, and it's going to go into the superior sagittal sinus, which carries venous blood. So all the junk that comes into the cerebrospinal fluid is going to be delivered to the venous blood in the superior sagittal sinus. So the cerebrospinal fluid comes through the central canal, goes through the subarachnoid space, but also goes through the ventricles inside these spaces, these hollow spaces within the brain. And where is it produced? So the ventricles, so the lateral, third, and fourth ventricle, they are lined with what we call the choroid plexus. And it's at the choroid plexus here represented in this reddish area, you see here also, where this is where the cerebrospinal fluid is produced. And I will explain. So it depends on the layer of blood vessels that are porous, and those blood vessels are contiguous to a layer of ependymal cells. If you remember, ependymal cells are a type of glia cells, and those ependymal cells form a barrier. So whatever comes out from the blood vessels cannot flow between the ependymal cells. It has to go into the ependymal cells, and then the ependymal cells is going to secrete, they are going to secrete whatever is there into the cerebrospinal fluid. But this, I will explain this better. So here, hopefully this will work. You're going to see the flowing of the cerebrospinal fluid, the circulation. Let's hope this works. So you see the lateral ventricles, the third ventricle, fourth ventricle, the aqueduct. And now you see the subarachnoid space. And now the villi, the arachnoid villi, and it's going to circulate, it's going to go out to the superior sagittal sinus. So it's produced at the choroid plexus. So we have arterial blood that decides or determines what is going to go into the cerebrospinal fluid at the choroid plexus. So you have arterial blood, which is the source of the components of the CSF, and then the nutrients are delivered and metabolites are removed, and they go through the venous blood, through the arachnoid villi. So if you see another view from the top, these choroid plexuses, you see here on the ventricles, the lateral ventricles, and also here. So all ventricles are lined up with these structures called choroid plexus, where the CSF is produced. Okay, so what makes up the choroid plexus? So the choroid plexus are made up of two important structures. First of all, the blood vessels, the capillaries that are present in these structures are highly permeable. That means that you have endothelial cells that have porous, have spaces in between, therefore allowing whatever is in the blood, in the plasma, to move out. But when they move out, so here's a blood vessel with what we call the fenestrated endothelial cells, so that the endothelial cells make up the walls of the blood vessels, and they are porous. So here's the blood. So that means that components in the blood can move out through these porous endothelial cells. We say they're fenestrated. Fenestra means window. Okay, so now you have these components of the blood vessel that are present in the blood. They move out to this stroma and they hit this wall of ependymal cells. And now these ependymal cells are not porous like the endothelial cells. These ependymal cells are attached to each other by tight junctions. Remember, tight junctions. So they are attached to each other in a way that nothing can go between them. Whatever is in the stroma, whatever is in this area coming from the blood, has to first get into the cells and then will come out. So the ependymal cells that form this wall, this barrier, we call it the blood CSF barrier. So it's made up of the ependymal cells, which are a type of glial cells, present only in the CNS, and they allow for filtration and excretion. So whatever gets in, it has to be pumped. The ions have to go through channels and so on. So they have to get into the ependymal cells and then they are secreted. So this space here outside, this whitish area, represents the ventricles, the empty areas in the ventricles. So you have here the choroid plexus. So you have the blood coming from the porous blood vessels, porous capillaries, hitting the layer of ependymal cells. Then the ependymal cells secrete the CSF that goes into the ventricles. So here you see another version of this with the capillaries that are porous and they allow nutrients and glucose and so on to come out, and waste will go in. And here's the ependymal cells, the layer of ependymal cells that forms this barrier and determines what is going to go out to the space in the ventricles, the empty space in the ventricle that's represented here by this white area. So the ependymal cells and the blood and the endothelial cells that make up the walls of the blood vessels make the blood cerebrospinal fluid barrier. So these ependymal cells are very, very important.
The question,
Yes.
so the ependymal cells decide how much of the fluid is going to go out into the blood?
If they decide how much CSF is going to be secreted, is
Yes.
that what you're asking?
Yes.
Yes. Yes. They are the determinants. Okay, so what is the circuit of the CSF? So it comes from arterial blood that is flowing through those porous capillaries. And when they reach the choroid plexus, so the components of the blood can leak, but they reach the ependymal wall, the ependymal cells that form the wall. But then it's going to be secreting the cerebrospinal fluid that now is going to circulate in the subarachnoid space that envelops the whole brain. You see this whole area through the ventricles and the central canal in the spinal cord. So the cerebrospinal fluid that flows through these areas then is going to reach the arachnoid villi. So these bodies, these structures here allow for the metabolites and so on to come out of the subarachnoid space and go right into the superior sagittal sinus, which is a blood vessel that contains venous blood. So you have the good stuff coming in at the choroid plexus, and the bad stuff to be removed through the arachnoid villi. And then it goes to the jugular veins. Now you can ask, so whatever is produced at the choroid plexus has to be removed at the arachnoid villi. If not, there's too much CSF accumulating in the brain. And this can lead to a condition known as hydrocephalus. So you want the amount of CSF that's being produced at the choroid plexus to be equilibrated with the amount of CSF that is removed at the arachnoid villi. Does CSF always a blood barrier, or can they exist without tight junctions? So, no, the tight junctions that unite the ependymal cells, that glue the ependymal cells to each other, are very important. If those tight junctions are not perfect, then you have a lot of leakage into the CSF that could damage the cells in the brain. Okay, so you see here magnification of the arachnoid villi that are in continuation with the subarachnoid space. And this is where the junk is going to be removed and going into the superior, into this blood vessel, the superior sagittal sinus. So the reabsorption at the arachnoid villi has to be in equilibrium, the same as the production at the choroid plexus. And here you see an image of a hydrocephalus. When there is no equilibrium between reabsorption and production, when there's too much production and less reabsorption, then the CSF will accumulate in the brain. You see here lots of fluid, and this fluid can act on the brain because the brain tissue is very soft, so it can exert pressure on the brain, and in a child maybe doesn't allow the full development of the brain. So this has to be immediately treated, and what happens, you have a shunt. This tube is placed in such a way that the extra fluid will go into the peritoneal cavity, to the abdominal cavity, so that the excess CSF is removed through this tube into the abdominal cavity to prevent the pressure on the soft tissue of the brain. So hydrocephalus. Okay, now we talked about this. What happens if there's less production and more absorption? So the opposite is not good either. If you have too little, then the brain is not, many problems can arise. The brain might not be receiving the right amount of nutrients. Also, you don't have that cushion that is provided by a full subarachnoid space filled with CSF. So that means that you don't have that protection. Okay, so we talked about CSF and the what we call the blood cerebrospinal fluid barrier. Now we're going to move on to the blood supply to the brain. We're going to be talking about the blood-brain barrier. It's different. Blood-brain barrier is what determines what's in the blood and gets into the brain. It's different from the CSF. So the brain weighs about 2% of our whole body weight, but it consumes a lot of oxygen. It's highly, highly active. And I wish that all this activity would lead to loss of weight, but it doesn't. You can be highly active with your brain, but you can still be very, you won't lose any weight. So brain exercise does not lead, unfortunately, to brain loss. I wish it did. So the brain receives 15% of the blood that circulates in our body. And it's very important for the blood, for the brain to be well nourished because it cannot store glucose. There's no glycogen in the brain. So it has to be continuously provided with glucose. So under starvation conditions, you can only go down to a certain level that does not cause a brain starvation because the brain does not store glucose or glycogen. Okay, so at the choroid plexus, we said that the walls of the blood vessels were very porous. They had these spaces that were called fenestras. So that molecules that are drugs can be delivered to the different areas of the body because we depend on these openings, these fenestras on the walls of the blood vessels. However, at the brain, not at the choroid plexus, remember? Choroid plexus, the capillaries also are fenestrated. Now we're talking at the other areas of the brain that do not include the choroid plexus. So the blood vessels there are not fenestrated. They form what is called the blood-brain barrier. That means that the endothelial cells have tight junctions. They form a barrier, just like what we talked about the ependymal cells, forming a barrier. Here, the blood vessels in the other areas of the brain that are not the choroid plexus, the endothelial cells are attached by tight junctions. Besides having this layer of endothelial cells attached to each other, the astrocytes also provide a barrier. So they provide a component to the blood-brain barrier. And then we also have pericytes, so that this barrier that is formed around the blood vessels in the brain establishes a mechanism that doesn't allow many toxins that might be flowing in the blood, in the capillaries. They cannot get out into the brain. So it's a mechanism to protect the brain from toxins and bad drugs. So you see,
Professor,
yes,
just to make sure, the blood vessels in the brain have a layer of astrocyte so that, unwanted stuff from the blood does not leak into the brain.
exactly. So the astrocytes, they are glia cells also, right? So they are a component of the blood-brain barrier. So the blood-brain barrier, this barrier, physical barrier, is made up of the walls, the endothelial cells that make up the walls of the blood vessels, plus the astrocytes and pericytes. They form a very thick barrier in terms of protection. So you see here in other areas of the body and also at the choroid plexus, this here represents endothelial cells that make up the walls of the blood vessels. And you see that there are spaces that we call fenestra. So drugs that are flowing in the blood, they can easily cross out. However, look at here at the blood vessels in the brain. Forget about the choroid plexus, other areas. You see that you have the endothelial cells, just like here, but they are connected by tight junctions. So there's no space, there's no fenestra here. Plus, you have the astrocytes that also contribute to this barrier, and you have pericytes, which are sort of a differentiation of smooth cells. of smooth muscle cells. So all these layers contribute for a very specific and efficient barrier that protects the brain tissue. So you see here smooth muscle cells, you see the astrocytes, the pericytes, and the endothelial cells connected by tight junctions. And here's the blood flowing. So to get from here into the brain stroma, right? It's very hard. You have the blood-brain barrier. So imagine how difficult it is to deliver drugs to the brain because of this blood-brain barrier. It's very hard because the drugs cannot go through, like here, through this fenestra. Okay, so what can go through and what cannot go through the blood-brain barrier? So the brain capillaries, the endothelial cells are connected by tight junctions. So lipid-soluble molecules and ions can go through. So nicotine, ethanol, heroin, all these can get into the brain very easily because they're lipid-soluble. So they can just cross the plasma membrane and get in. Water-soluble molecules cannot cross the blood-brain barrier. They have to go through transporters, glucose transporters, amino acid transporters. The astrocytes are glia cells that are major contributors to the integrity of the blood-brain barrier. So it's thought that many neurodegenerative disorders, the blood-brain barrier disintegrates. And if the blood-brain barrier disintegrates, then you can have a lot of toxins getting into the brain. So you get the breakdown of the blood-brain barrier, can be a major contributor to neurodegeneration, to the death of the neurons. So an interesting, it's very hard, as I told you, to deliver drugs to the brain. And for example, for Parkinson's disease, where you know that the dopaminergic neurons are those that degenerate, the patients cannot be treated with dopamine because dopamine is not lipid soluble and cannot cross the blood-brain barrier. So they are treated with L-DOPA, which is a precursor of dopamine. And L-DOPA can cross the blood-brain barrier. So there are certain areas in the brain where there is no blood-brain barrier, right? We've been talking about the choroid plexuses. You know that the choroid plexuses that are located at the ventricles, at the brain ventricles, there is no blood-brain barrier. And there are other areas in the brain where the blood vessels do not contain a blood-brain barrier. And those areas are known as circumventricular organs, or CVOs. Circumventricular organs. Some of them are the choroid plexus, but there are other that are located near the hypothalamus, the pituitary, and the pineal gland. So why is it that at these areas of the brain, there is no blood-brain barrier? Why? Because these parts of the brain, the hypothalamus, pituitary, and pineal gland, they produce hormones. And you want the hormones that are produced by these glands to get into the blood and to be distributed so that they can have an effect on the target organs. So blood vessels near the hypothalamus or the pituitary or the pineal gland, there is no blood-brain barrier. So they are also part of the circumventricular organs. So circumventricular organs are areas or organs in the brain where there is no blood-brain barrier. So you have the choroid plexus, pineal gland, posterior pituitary, and hypothalamus. So why can't we deliver drugs to the CVs? Yes, very good idea. But these circumventricular organs are not present in all areas of the brain, right? So if you want to deliver to the frontal cortex, there are no circumventricular organs there. So some drugs that are targeted to areas that are close to the circumventricular organs, they can be delivered that way, but many cannot because of the blood-brain barrier. Very good comment. Okay, so how do viruses get into the brain, right? If you have this blood-brain barrier, how can viruses get into the brain? How? They get in through the circumventricular organs when there is no blood-brain barrier. So you see here the HIV gets into the brain through these openings, the fenesters at the circumventricular organs.
Professor,
Yes.
the hypothalamus, the pituitary, the pineal glands, all of these would fall under the category of CVOs, right?
So can you say the last part? I didn't hear.
Sorry, all of these places where the blood-brain barrier is not present, like the hypothalamus
Yes.
and the pituitary gland, all of these would fall under the category of CVOs. Are they all CVOs?
Are they all what?
Circumventricular organs?
Yes.
Oh,
Yes. They're all CVOs. Is that what you were saying, CVOs?
yes, yes. Sorry.
Yes. They're all CVOs. Yes. Absolutely. Because, the hypothalamus makes many hormones. Like, remember oxytocin, right? So if you have blood-brain barrier there, what would be the point? It could not get into the blood and it could not reach the uterus, right? The posterior pituitary makes ADH, antidiuretic hormone, and oxytocin are both produced by the hypothalamus, and then they go to the pituitary, and from the pituitary, then they go into the blood vessels for the circulation. Antidiuretic hormone is used to prevent the excretion of water. So to maintain certain level of water in the body, this is anti-diuretic hormone. So it's produced by the hypothalamus, goes to the posterior pituitary. From there, it goes all the way to the kidneys. So you cannot have blood-brain barrier here. The same for the pineal gland that produces melatonin. So these produce hormones that are required to be secreted into the blood so that they can be distributed and reach their targets. Okay, so now we arrive at the major areas of the brain. So we're going to study what are the functions in these specific areas of the brain. So you see here the cranium and how nicely arranged and tucked into this small area of the cranium, all these different parts of the brain. Here you see the spinal cord, the hindbrain shown in green, and the cerebellum. So the hindbrain is made up of the medulla and the pons. See here in green, hindbrain. The pons and the medulla, they contain the centers for control of heartbeat, circulation, respiration, hunger, satiety, and so on. So remember homeostasis. Then you have the midbrain. The midbrain contains the hypothalamus and thalamus, midbrain in blue. Then you have the forebrain. The forebrain is the largest area of the brain. Here, this area is known as the corpus callosum. The corpus callosum is made up of myelinated axons. And this is very important because it allows the two hemispheres, the right and left hemisphere, to communicate. So for the electrical current to go from one hemisphere to the other, it relies on the processes, the fibers, the tracts that are located in the corpus callosum. So if you want to separate the two hemispheres, you have to cut the corpus callosum.
Professor, I'm sorry, I thought you said that hypothalamus was part of the midbrain, but why is
No,
it red
it,
color here?
it isn't. It's part of the cerebrum. Sorry. Mistake, my mistake.
Oh, sorry. So the midbrain is only the thalamus?
Yes, it's basal ganglia and substantia niger. You will see. We will describe the midbrain.
Oh, thank
You see here the pituitary gland is also part of the forebrain. Okay, so what happens when we do a simple exercise like playing tennis, right? So the ball is coming to our court. We need to see the ball. Where is it going? Is it going to the right? Is it going to the left? What is the part of the brain that's going to make us able to make that prediction? That's the visual cortex. It's located all the way here. At the same time, we need to make a decision. Which muscles are we going to use? The legs, the right leg, the left leg, right arm, left arm. So we need to devise a program for movement. And that's the premotor cortex, located here in the parietal lobe. That has to come into action. Then we have the amygdala, another area of the brain that allows us to, allows the body to adjust in terms of heartbeat, respiration, right? Because we're going to be running. So the centers that regulate uh, heartbeat and respiration rate and so on, the amygdala comes to be active. The limbic system, that's emotion. So that's coming into action to say, well, we want to hit the best possible shot. So that's the emotion, that's the limbic system coming into action. Then the spinal cord is going to conduct the electrical, is going to determine where are those electrical currents going to the specific muscles, right? The effectors. Then basal ganglia. Basal ganglia is located in the midbrain. Okay. Basal ganglia, remember what ganglia are. Ganglia are groups of neuron cell bodies that are not in the gray matter. So basal ganglia are the location for learned movement. For example, learning how to ride a bicycle, learning, you have many lessons to learn how to play tennis. All this knowledge is stored in the basal ganglia. Cerebellum, this area here, allows for fine tuning. And then the hippocampus, memory, so that you can remember this amazing shot that you produce. So you see that a simple action of hitting a ball with a tennis racket, how all these different areas of the body come together to produce an optimal effect, an optimal response. So, what part of the CVO is affected by alcohol? Destroys our balance. So, the cerebellum is involved in balance and other areas. So, the limbic system is emotional. So, it makes you want to hit the best possible shot. Okay. Okay, so we are going now to focus on the cerebrum. It's the site of intelligence, so abstract thought. It allows us read, write, speak, math, music, memory. So creativity. AI is not there yet. So how is it that we can generate new thoughts? Creativity is very, very important. And it's the largest area of the brain, the cerebrum. So the cerebrum is divided into two hemispheres. right hemisphere and left hemisphere. And you see here that the two hemispheres are connected by the corpus callosum. So the corpus callosum contains only white matter. So white matter means myelinated. So these are the axons that allow for the information, I mean electrical current, to go from one hemisphere to the other hemisphere. And you see that the two hemispheres are separated by these fissures. And the whole brain is so big that it had to be folded to fit in the cranium. So it has fissures, which are and sulci. So the folds, gray matter grows faster than the white matter. And that's why you have these gray folds. And the grooves are these, let's say, valleys that are generated between the folds. If they are deep, like these here, they're known as fissures. If they are shallow, the ones like here, they're known as sulci. Okay, so we have two hemispheres, and their function, their actions are very different. For example, the left hemisphere, so the two hemispheres, they control opposite sides of the body. So the right hemisphere will control the left side of the body, and the left hemisphere controls the right side of the body. And they have different functions, as you will see. Okay, now let's focus on the gray matter. Remember that in the brain, the gray matter is on the outside, and its thickness is about 2 to 3 millimeters. And it has 6 different layers of neurons. So you have the pyramidal cells, then satellite cells, large pyramidal cells, and so on. So this here, these 6 zones have different neurons, different types of neurons, and they are located, the cell bodies of these neurons are located in the gray matter. So billions of neurons. The white matter the white matter here contains the tracts. Remember what the tracts are. The tracts are the fibers, the axons that are myelinated and that are located in the CNS. So these tracts connect one hemisphere to the other. They connect different areas within one hemisphere, and they connect the hemispheres with lower areas of the brain. So here you see one of my favorite scientists, which is Santiago Ramon and Cajal. He was from Spain, and he won the Nobel Prize in 1906. So he spent hours and hours and hours. His father was a physician, and he liked to look at preparations from the brain, and he stained them with this black dye, which is called the Golgi stain. And the Golgi stain was distributed throughout the cell bodies and the processes of these neurons. And he spent hours and hours looking under the microscope and drawing all these different types of neurons present in the brain. And there are museums that actually show his drawings. Santiago Ramon and Cajal. He was a very mischievous child. He didn't like school, and he did a lot of mischief things in a good way. Nowadays, we don't need to use Golgi stain. We have different ways of looking at the brain. Actually, there's a way of destroying the stroma of the brain so that it becomes transparent. And this process is known as clarity. So you destroy the connective tissue and so on, so that the brain becomes transparent. And all you can see are the neurons. Let's see if this works here. Why can't I reach this? Yes. Let's see. Oh. sorry about, okay. I didn't want to have an ad. I'm very sorry. But now what you're going to see is, so the stroma of the brain is gone. What you see is the neurons, the processes and the cell bodies of the neurons, no connective tissue. And you can see how incredible all this structure is and how hard it is to study and unbelievable. Okay, let's move on. Okay, now the brain, we have different, what we call different lobes. And why do we divide the brain into different lobes? Because each lobe sort of encases specific functions. So we have the frontal lobe, which is the intellect part of the brain, and it's in the front, as its name indicates. Opposite of the frontal lobe, all the way in the back, is occipital lobe. This is where the visual area is located. The parietal lobe stays between the occipital lobe and the frontal lobe. You see the parietal lobe. There's a lot of different functions. Here you have the sensory cortex, right in the limit of the boundary of the parietal lobe with the frontal cortex. You have the temporal lobe right here. Language is located here, olfaction, taste. And then, so these are the four major lobes. And then we have the insula, which is a lobe, an area that is deep inside the brain, where the language center, the Broca and Wernicke area are located. So what are these lobes? What am I talking about? So these are areas like the map where, so any sensation that activates the visual, the photoreceptors in the retina, the action potentials are going to be delivered to the occipital lobe. Any sensation, for example, from the taste buds, those action potentials are going to be delivered to an area here in the temporal lobe that has the gustatory area. So that's what I mean. So these lobes are going, or the brain is divided, the cerebrum is divided into these lobes because they indicate where the different action potentials generated at the periphery, where they are going to be conducted. And this is what allows us to differentiate different sensations, is where the action potentials that they trigger, where the action potentials are going to be, are going to end up, where they're going to be delivered. So now, how about the white matter? So the white matter are the fibers, the axons that are myelinated and that allow for communication among the two hemispheres. Those are known as the commissural fibers. So they allow for the right and left hemisphere for the action potentials to travel from the right to the left hemisphere and vice versa. Those are not located in corpus callosum. Then you have white matters that allow action potentials to travel within one hemisphere. This would be the association fibers. And then you have fibers or tracts that go down from the brain to the spinal cord. And these would be the ascending and descending fibers for projection. So you see that these tracts, these axons, allow for communication between two hemispheres, among different areas of each hemisphere, and then down to the spinal cord. This is the corpus callosum. And here's the basal ganglia that are located in the midbrain. So basal ganglia are groups of cell bodies of neurons that are located within the white matter. So remember, the gray matter is on the outside. The middle is supposed to be all white matter, but there is some gray matter. in the middle that makes up the basal ganglia. And these basal ganglia, you see here some names: the putamen, caudate nucleus, globus pallidus, and subthalamic nucleus. So these are brain and substantia nigra. So these are cell bodies, groups of cell bodies of neurons that are located within the white matter, and that's why they're gray. They make up basal ganglia. And what are they dedicated to? Learned movement and subconscious control. Okay, I think I'll stop here, or maybe I'll go just the limbic system. Well, a few more minutes. So the limbic system is not dedicated to a specific lobe. So different parts of the brain, of the cerebrum, contribute to the limbic system, which is the emotional brain. So you have the cerebrum, diencephalon, and mesencephalon. They contain components that make up the emotional brain, the limbic system. So you have the hippocampus, amygdala, frontal cortex, and cingulate gyrus. These are all part of the limbic system. And how do we know that this area, especially the frontal lobe, that is dedicated to emotion? So there's a very interesting story that happened in 1848, so 19th century. There was this railroad worker. His name was Phineas Gage. And Phineas Gage was a manager of the company that was building this railroad in Vermont. And he was a wonderful person, always on time, very kind to everybody. And one of his functions was to put down explosives to break up the rocks so that they could put the tracks, the metal tracks, iron tracks, to build the railroad tracks. So one day he had explosives. He was just distracted for a few minutes, and he switched on the explosive too early before he left the area. So this one of these iron bars flew through his head, going through below his left eye and coming out through the frontal lobe. So you see here, this is the cranium, and you can see very well this iron rod that went through his skull and affecting the brain, the soft brain tissue. So what happened to Phineas Gage? Well, he could walk. He was fine. He fell a little bit, but he was able to get up. He went to the doctor, and the doctor cleaned and so on. And he rested for a while. And then after a few months, he came back to work. He was fine. But what happened? Phineas Gage was no longer that nice person, that nice manager. He was mean to everybody. He was never on time. And his personality totally changed. And it changed because of the damage to the frontal lobe. So this is one of the first episodes or events that led us to understand that emotion is housed in the frontal lobe. And I think I'll stop here. So, final. Did he realize his change? I think so, because he had to stay back home. Did he remember? Yes. And eventually he was fired because he was persona non grata. So have a great Thanksgiving, and I'm going to try to post all the lecture material, and I will see you a week from today. Goodbye, everybody. Bye-bye.